Okay, so the next question that comes to mind is a question of rather the relationship between genome size and inducing function. Does genome size automatically induce changes to function? That means that does the fact that, let's say, we would add or double the amount of genes and DNA to the human body, human species, would that necessarily change function? Which is an interesting question. So DNA can be represented in mRNA if it is hybridized with RNA. And 1% of DNA acts as a template for mRNA, right? So the higher the organism, the more genes are expressed, especially if you look at a certain organ as the brain. The brain actually has the highest rate of gene expression in the body. So about 50% of the mass of mRNA in a cell is from a single mRNA type. So that's very, very interesting, right? Because you have one mRNA type that is 50% of the cell's mRNA mass, and all the rest account for 50%. An additional 15% on top of those 50 come from up to 8 other mRNA types. So right now, 65% of mRNA mass in the cell comes from 9 types of mRNA. Just to clarify, 50% comes from one type, and an additional 15%, together 65, come from 8. So 8 plus 1 is 9, and 50 plus 15 is 65. Okay, and the residual 35% of mass comes from up to 13,000 different mRNA types. So here's the recap. One type of mRNA equals 50% of the mass of mRNA in the cell. An additional 8 types add 15% of that mass, and 13,000 mRNA types build up or add the additional 35%. So if you connect the dots, about 13,009 types of mRNA equal 100% of the mRNA mass of a cell. So let's say that we could open a cell, take off the upper part, and start counting mRNAs of the same type, kind of tagging them from the same type. So we would say that the more you have of the same type, the greater its abundance. Okay? And the abundance of a certain mRNA can be calculated if the total RNA mass is known. Because as you know, if you know the RNA mass, you know what to expect at the mRNA level. Okay, so we can divide basically mRNA into 2 classes. We have about 100 different types of mRNA molecules that are represented in between 1,000 to 10,000 copies, and they are equal to 50% of the total mRNA mass. Okay, and we have 50% of the total mass is represented by 10,000 types of mRNA of less than 10 copies each. So we have, in conclusion, we have small, we have mRNAs that have only 10 copies each, but there are about 10,000 of those. And we have mRNAs that have between 1,000 and 10,000 copies, but we don't have much of those. The second type, meaning those with only 10 copies, are what we call scarce mRNA or complex mRNA, meaning that they have special function. You almost relatively to the total amount of types and molecules of mRNA, they will be really hard to spot in the cell. The expressed number of genes, especially the somatic ones, can vary between tissues. So we're talking about cells and we're talking about tissues, but we have to understand that every tissue has a different level of expression, even for the same genes. Okay, so the expression has to fit the needs of the tissue. And of course, just a very quick reminder that we go from cell to tissue to organ to system, right? So the needs and the function of the cell has to serve the needs of the tissue and thus the needs of the organ and even the system. So here's a question: how many genes overlap between two tissues when compared? It's interesting, right? Is every tissue type going to use a completely unique set of genes, or are there genes that are expressed in both tissue types, and to what extent? So in order to answer that question, we need to look at the transcriptome. As we've talked before, we have mRNAs of high abundance and mRNAs of low abundance, right? And the answer is going to be probably evident, and it probably lies in the high abundance molecules. In addition, I want us to remember that there is a difference between the percentage of mRNAs and the number of expressed genes. So it's not a ratio of 1 to 1, there's a difference. So in order to know how many genes are expressed, we must focus on the scarce mRNA of the cells. Logic dictates that the more linked the tissues are in function, the more common expressed genes they will have, including scarce mRNA. So that makes sense, because if a gene codes for a certain protein of a certain function, and that function is needed in both tissues, there is a better chance of finding that gene expressed in both tissues. Does that make sense? At the same level, let's say that forks are being used by humans. So the chance of me finding a fork in my home and your home and someone in Israel, someone in Qatar, and someone in Portugal is extremely high, because the fork is used. Okay, so fork is a tool of function with a certain function, thus the expression of its use will be conserved between different areas, right? So that's the analogy to the body. A protein that has a very common day-to-day and important use or function will be found in both tissues, and thus the gene will be expressed in both. On the safe side, it will be extremely safe to say that at least 10% of the genes that are expressed are different between two tissues. If you extrapolate, that means that about 1,000 genes are expressed and are responsible for the essential functions of all cell types in mammals. So we're talking about 1,000 genes that are shared in expression between cells and tissues. These cell common functions are called housekeeping or constitutive functions. And the gene gets the same name. So if a function is regarded as a housekeeping function, the gene will be called a housekeeping gene. But if it is not one of those 1,000, that means that it is luxurious. You don't have to have it, but you want to have it, and it's nice to have it. So we have luxury genes, and those luxury genes are those who are needed for specific function of a specific cell. Okay, so they kind of differentiate between cells and between tissues accordingly. You're referred to a movie, enjoy the movie. Okay, so now we're going to talk about non-Mendelian inheritance. And what has been found actually that plants have organelles that have DNA as well, what we call non-nucleus DNA. So inheritance is mostly represented by the lack of association between two things. The segregation character and the meiotic spindle. Okay, you're referred to a movie, that should clarify that. And the second thing is the segregating character and mitotic spindle. That is called also somatic segregation. Okay. I don't want to go into that too much in order to avoid the need to go over the cell duplication cycle, but if you want to, you can look it up and learn all the cycles there are. So that's not emphasized in this course and in the test accordingly. Okay, so a mitochondria might inherit from both male and female with different alleles, right? Producing an unbalanced daughter cell representing only one parent's information. So the idea is extremely simple. If the inheritance is not balanced in the sense of dominance and the ability to affect, we might find out that one parent's influence of genetic matter and information is greater than the other. A boy or a girl or both are born in the hospital, and what is the first discussion that starts? Yep, who do they resemble? God forbid they resemble somebody from the other side of the family, right? So, and the truth is that some features are very easy to identify at this point, and some are going to change. For instance, eye color. So I wouldn't start a war over who does the child resemble, but naturally that's what happens, right? Oh, he has your nose. Oh, he has your mother's eyes. Oh, he has your father's chin. No, he doesn't. Yes, he does. So we see that we can talk about one parent's genotype is either lost or not expressed, and that is a recipe for problems. So when that happens, when one of the parents' genotype is lost during the inheritance of the genetic information, it is called uniparental non-Mendelian inheritance. So the Mendelian inheritance theory says that we inherit one copy from every parent, and they are both expressed or have the fair chance, the balanced chance of equal chance of being expressed, right? But this is not what we're talking about. We're talking about here about one parent's genetic information loss. So this is a bit different. And then there's a phenomenon called, an interesting one, by the way, and a very important one, obviously, that I'll explain, called maternal inheritance, which by far is stronger if a daughter cell is balanced. So through evolution and selective forces, and according to what we see in nature, maternal information is stronger than the father's information, just so you know. For instance, one example would be when you look at the possibility of inheriting, yes or no, twins. If the mother's genetic information includes twins, there is a way higher chance of the twins actually being born. Such a behavior contradicts the classic Mendelian genetics, whereas copies of both parents are presented in the daughter cell, right? So it's unusual, but it does happen. Right, we'll stop here and continue in the next video.